In today's session, we will begin with the next event, that is pollen pistil interaction. This specific event will begin after the pollination and before fertilization. As we have already completed androecium, a male part, gynoecium, a female part, with their members stamen, carpel, having fertile parts anther and ovary, showing microsporogenesis and megasporogenesis to produce microspore or pollen grain, that is male gametophyte, and megaspore or ovule, that is female gametophyte. The next event is transfer of these gametes, as both are non-motile, and here in flowering plants this transfer we call pollination, which is the transfer of microspores from anther to the stigma of the same flower or among the two flowers growing on the same plant or two flowers growing on different plants which belongs to same species. We already discussed the agents involved in pollination. We already discussed the conditions for self-pollination and cross-pollination. Now in today's session, I will explain what exactly happens when the stigma receives pollen grains. The first step, when pollen grains deposits on the stigma should recognize their own pollen grains. For this, stigma will secrete a sugary fluid named as exudate. Now, the pollen grains of their own species can absorb this exudate. Pollen grains of other species unable to absorb. So, the viable pollen grains are the one who can identify that exudate and start imbibing it, while non-viable pollen grains unable to absorb this fluid exudate. Now the viable compatible pollen grains absorb the exudate, imbibe, and now the next step begins. In step 2, as we already know that pollen grain is two-celled structure. It has vegetative cell with big irregular shaped nucleus and generative cell with small nucleus. It is covered by two layers, exine with the germ pores and intine, which is continuous. Now here in the second step, vegetative cell start germinating through the germ pore as a tube, and this we call pollen tube. As this pollen tube start penetrating through the tissues of stigma and start entering in the style, generative cell along with its nucleus is shifted in this pollen tube. So, you can imagine now the pollen tube along with the generative cell start entering in the style. This pollen tube now reaches the ovary. Now in the ovary, megasporangia or ovules are already mature. Each ovule or megasporangium is having a funicle, hilum, chalaza, integuments with an opening micropyle enclosing a nourishing tissue nucellus with 7-celled 8-nucleated embryo sac, out of which 3 cells towards micropylar region forming an egg apparatus having egg and on either side synergids with filiform apparatus. Central cell with 2 polar nuclei, and towards the chalazal end there are 3 antipodals. Now this ovule is mature ovule, anatropous in condition. Now as pollen tube enters the ovary, there are 3 options. Either the pollen tube penetrates through chalaza or through integuments, or they can enter through micropyle. It depends on the position of ovule. This entry is classified as chalazogamy, pollen tube entering through chalaza. If it enters through integuments, it is called mesogamy, and when it enters through micropyle, it is porogamy. Now in this, let us consider porogamy, means pollen tube will come near the micropyle and enters. One of the synergid with its filiform apparatus will receive this pollen tube. Meanwhile, the generative cell divides, giving two male gametes. As the pollen tube enters in one of the synergid, it disintegrates, pollen tube ruptures, and two male gametes are released inside. Out of these two male gametes, now one will try to fuse with the egg and one will try to fuse with the central cell. Till here, whatever events we have studied is called pollen-pistil interaction. These events includes the steps after pollination and before double fertilization. Let us revise once again. First step, deposition of pollen grains on the stigma. Step 2, stigma secretes a sugary fluid exudate to identify viable compatible pollen grains. Step 3, compatible pollen grains absorb this exudate, imbibe. Step 4, vegetative cells start germinating through one of the germ pore as a pollen tube and it start carrying the generative cell within it. Next step, pollen tube start penetrating through the stigma and style and reaches the ovary. Next step, in porogamy, through micropyle pollen tube enters, generative cell divides giving two haploid male gametes. One of the synergid with the help of filiform apparatus receives the pollen tube. Tip of the pollen tube ruptures and two male gametes are released freely. Now the next event we call double fertilization. This we can cover in the next session. Hope it is followed clearly. So in the first session we have completed parts of typical flower. In second session, androecium, stamen, parts of stamen, young anther, mature anther, microsporogenesis, structure of pollen grain or male gametophyte or microspore. Next we have completed gynoecium, members carpels, parts of carpel, ovary, it is megasporogenesis, structure of mature anatropous ovule. Then we have completed pollination, under which types autogamy with conditions, allogamy which are of two types, it is geitonogamy and xenogamy. Their conditions based on the agents involved, pollination is considered as anemophily where wind is involved, zoophily where animals are involved and hydrophily where water is involved. After pollination, in today's session I have covered pollen-pistil interaction, so that only the best one, healthy pollen grains are allowed to germinate, enters along with the two male gametes and now they are ready for double fertilization. Okay, with this I will complete today's session. Thank you very much.